Hey there, 7th graders. Mr. Smith back in the bubble, here to tell you about our new inquiry project for 7th grade science called Ecology in a Bottle. We're going to be designing a quick experiment with materials you can find around your house or pick up at any big box store like Walmart, Target, or Home Depot. I strongly encourage you to involve your parents, siblings, or anyone else in your household in this project. It'll be a lot of fun to go over your observations and share what you've been learning in class with those people in your home. This project is optional, meaning that while you won't be penalized for not completing it, you will be rewarded with 10 points in your projects and exams category on Infinite Campus, in addition to getting a deeper understanding of what ecology and populations mean. So with that, let's take a look at our experiment. What's this project about? Well, it's going to give students the opportunity to observe ecological changes in a short period. Typically, ecological studies take anywhere on the order from weeks to years to complete, but with a small environment that you're containing, it'll be a lot shorter and you'll see a lot more dramatic change than you might in a larger ecosystem. It's a hands-on activity. You're going to be exploring what we learned in our ecology unit now, populations and resources, and in our last unit for the year for 7th grade science, matter and energy and ecosystems. So going through this project is really going to give you a good idea of what to look forward to and apply some of these concepts in a real-world experiment. You can design your own bottle, so that means that you're going to have the opportunity to be creative, stretch those kind of creative muscles out, and that way you can have a little bit of fun with this. Or if you're not confident in how to design a ecosystem inside of a bottle, you can follow my directions, and that'll give you a good place to start for building your own ecosystem. This project will be 6 weeks long, starting as soon as Wednesday, 4/15/2020, so that's the Wednesday after we get back to school, and ending Wednesday, May 20th. That's going to be 5/20/2020, the second to last week before the end of the school year. Building your ecosystem should take maybe about an hour or so, and then your daily observations should take between 5 to 10 minutes, long enough for you to write in what you need, get your observations down on paper, and then moving on. This is a project that I want you to be able to do pretty quickly and have something to do every single day for science. These are some of the materials you'll be needing if you're interested in completing this project. First, you're going to need 4 plastic bottles. 20 ounces or whatever size you have available will be really good. I find the 20-ounce bottles to be just the right size. If you want to go bigger, go bigger. If you want to go smaller, you're going to have to be a little more delicate, but it should still work as well. You're also going to need a pair of scissors, a box cutter, or some other cutting implement. Make sure that if you're not comfortable using those materials, you contact your parents or whomever is in your household who can help you and get you through this project so that you're not imposing a danger to yourself. You're going to want some tape as well. Packing tape is the best because it's completely made of plastic and it won't absorb water, but any tape will do, including masking tape that you might already have at home. You're going to want a drill or a nail or some other piercing tool like an ice pick. You're going to be using that tool. If you're not familiar with how to use those tools or you're nervous about using them, again, enlist the help of someone in your household who is comfortable with those tools. I don't want anybody cutting their hands trying to get through this inquiry project. You're going to want safety glasses. Whenever we do a lab experiment, we're going to wear our safety glasses. It keeps our eyes safe, especially when we're building our ecosystems. We want to make sure that we're not getting plastic in our eyes, possibly cutting our corneas or something dangerous like that. Cutting board. I put this in here because I don't want you making your cuts on the kitchen counter or in your tables and your dining rooms. Let's save that furniture and keep your mom from yelling at you by using a cutting board and being safe whenever we can be. You're going to want pliers or a handheld vise. These is just going to help you hold on to the plastic. That plastic can be sharp when cut, so you want to make sure that you're not handling it with your bare hands where you can cut yourself or get a serious injury. You're going to want some soil as well. You don't need to get like a giant 10-pound bag of soil. Any amount of soil will do. Just a few handfuls will be enough. You can find that in your yard, or if you're out on a walk, just grab a couple of handfuls of soil as well. Seeds, you might want several different types depending on what you choose for your experiment, and I'll go over that in just a minute, where you can find seeds and what seeds grow best. You're going to want some water, tap water, bottled water, it doesn't matter. You just want water because it's basically going to be growing a garden inside of a bottle. And you're going to need a string or a candle wick, something made out of cotton. A plastic string or rope isn't going to work because plastic doesn't absorb water, so you want something that's going to absorb water for this experiment. So where to source some seeds? You can get seeds basically anywhere. Garden centers will definitely carry them. Big box stores will certainly have them as well, and they'll have a lot of variety in the seeds that you can pick up, such as flowers or other garden plants. If you don't want to go that route, or you don't want to spend the extra $1 or so on a bag of seeds, you can also get free seeds from your produce. Green peppers and bell peppers in general are really solid sources of seeds. You plant the entire core of the pepper in the ground, and those will typically sprout within a few days. Citrus seeds are also a really good option because those sprout very quickly. Potatoes and other tubers can be buried directly into the ground and will start growing a little bit more slowly than some of the other plants, but they will still grow, and they are really excellent. If you are going to go with a potato, make sure that you have a container that is large enough for a potato. I don't want you trying to put a giant Idaho potato inside of a 20-ounce bottle. And the one that I like to use are actually chives, or green onions. If you cut those up, the bottom section here at the bottom white section with the roots will actually sprout more chives, and this grows really quickly. Before you begin, here are some questions you're going to need to answer before you start your project, and even before you build your ecosystems. Consider what your hypothesis will be. Think of this lab kind of like our science fair projects. You're going to be developing a hypothesis and then experimenting through the lab itself. We're going to do a really excellent example here is a contained ecosystem with sun exposure grows plants more slowly. Remember, a hypothesis isn't a question, it's a statement about what is going to happen. What's your independent variable? If you don't recall, independent variable, it's the thing in an experiment that you're going to change. It's the thing in an experiment that is going to be different from your control or the thing you're not changing. Your dependent variable is the thing that you're measuring. It's whatever you're looking at. In the example I gave above, I'm going to be looking at the rate, or how fast those plants are growing day to day. And then what are your constants? What are some of all the things that you need to stay the same in this experiment to keep it constrained? You don't want things getting out of control. You want to make sure that everything is accounted for, and you want to make sure that all the things aside from your independent variable are exactly the same. So what's the goal for this experiment? Your goal is to build 2 ecosystems in a bottle that are identical in every way but one. Remember, that's our independent variable, the thing that we're changing. Otherwise, your bottle should be exactly the same. You will observe both ecosystems, taking notes of changes in the water, soil, seeds, and any other changes you can observe. Maybe the plastic is changing. Maybe there's algae growing in the water. That's up to you to determine what is an observation that you need to take a note of. Create a document summarizing your experiment's results. I'm going to have a document available for you that you can fill out, or you can do your own. As long as you have a full summary with all the pieces I need, that should be perfectly fine artifact for this experiment. So, if you're going to build your own bottle, or you have a design that you want to try, you can skip this section of the video. Otherwise, I'm going to move right into explaining to you how to build your own ecosystem in a bottle with just a handful of materials from your home. Step 1. Cut each of your 4 bottles about 3 inches from the cap. Adjust this distance if you have a larger or smaller bottle. Use your best judgment. Again, from the cap, you're going to cut about 3 inches away. I found that was about the right distance for the experiment that I'm conducting at home. Step 2. Drill a hole through 2 of the bottle caps. Remember, we're going to be building 2 of these, so you'll be making 2 copies of basically everything. If you're, again, not comfortable using a drill or nails or anything like that, please contact an adult who can help you with that, and they will be happy to put a small hole through the cap. The cap hole shouldn't be very large, just enough to pass that string through, because our next step is to pass that cotton cord right through that hole in the bottle cap. Then you're going to take that bottle cap and screw it back onto the bottle tops. So at this point, you should have 2 bottle tops with 2 bottle caps on top of them, with 2 strings running through each bottle cap. Fill at least 1 bottom half of the bottle with water. If your independent variable is the amount of water, this is the time where you would change the amounts of water that you're using, but otherwise, I find filling the bottom half of the bottle with about halfway water works pretty well for this experiment. Step 5. Place the top halves of the bottle into the bottom halves with the string hanging into the water, if the water is present. Just like the picture is showing you here, you want to make sure that that string is down in the water so it's wicking up moisture up into your second bottle. Step 6. Fill the top halves of the bottle with soil. So you're going to take your trowel here, or your hands, whatever you're using, and just dump some soil up into this top bottle, just like it's showing you in the picture. I find that about halfway up this bottle, maybe a little bit more, makes for a really excellent platform to bury your seeds. The next step is obviously to bury your seeds in the soil. Make sure that they're just below the surface. You don't want them too deep. You don't want them just sitting on top either, otherwise they won't take root. Step 8, water your soil. This is kind of an important step because you don't want to overwater your soil. So if your soil is damp, that's good, but if it's soaking wet and you can see water kind of coming out of it, or if you tip it on its side and water pours out, that's too much water. You got to give it a little time to dry out and start over. Step 9, we're going to tape the top onto your ecosystem, and now you're sealing it. Your ecosystem is now closed with the tape wrapped around the top, so now nothing can get in and nothing can get out. The only thing that's coming into your ecosystem should be water from the lower bottle that you placed earlier. Step 10, place your bottles in the sun for 6 to 8 hours a day. This is really important. Plants do need a lot of sunlight to grow. And for this experiment to be successful, you're going to want to put them in a sunny windowsill, outside in your porch, or anywhere where they're going to get a lot of sun, preferably between 6 and 8 hours a day. If the sun energy is going to be your experimental variable, then that's something that you'll have to look at yourself and change that accordingly. Step 11. Label the system that is normal, the one that was made exactly like this and was following these directions, as control. You're going to label the second system with the changed element, or your independent variable, as experiment. And you want to make sure that you keep those well labeled so you don't get them mixed up later. What are some independent variables you could consider? Really, anything will work, but here's a couple ideas that I thought of things that you could change between your control and your experiment bottles. Remember, you're only doing one of these as your independent variable. You can add an insect or worm that you've captured. If you're adding an insect or worm, I recommend that you double check to make sure that that insect can survive on only plants and doesn't eat any meat, because otherwise it'll die pretty quickly. Plant different seeds. This one's pretty simple. Pick up 2 different kinds of seeds and see what happens. You can use different amounts of water. Find out what happens in a more arid or drier climate. Different amounts of soil. Maybe some of the soil gets eroded away, and you can show that by having a bottle with less soil in it. You could use different size containers. Maybe you have 2-liter bottles and then 2 20-ounce bottles, and those form 2 ecosystems of different sizes, and see how that affects things. So making observations. This is something we talked about in the last few weeks in class before we went to our digital curriculum. Remember, when we're making observations, we want to make sure that we're making good scientific observations, not just blurting out the first thing that comes to mind. So make sure you're using some of that science vocabulary and making observations daily and at the same time of day if you can. When taking notes, remember that what makes a good scientific observation is never using subjective terms. Terms like, oh man, this looks weird, or this looks gross. Those aren't words that really tell us a lot about the experiment. Remember, you're trying to describe this to someone as though they are not there with you and they don't know what you're looking at. On that same thread, saying it looks like dirt or it looks like milk, it doesn't really tell us a lot, and especially consider that you might be having an audience of people who've never drank milk, who don't know what milk looks like, who don't know what the dirt in your area looks like. So you want to make sure that you're being as descriptive as you can, using descriptive language and not just saying it looks like other things. Things that make for good observations: you can say it appears wet or it appears dry. You can also say things like you notice condensation or you notice evaporation. Those are really good observations. You can take note of the temperature of the soil, water, or air in the ecosystem. And you can check the weather conditions on that day. If you had a particularly cloudy day, that might be something you want to note in your observations for the day. For your documents, these are the things that you're going to be submitting to get your credit for this assignment. Create your own note catcher or use the one in Google Docs that I'm attaching to this assignment. If you're not sure where to start, the Google Doc is a good place to look for ideas if you want to build your own as well. When you notice a significant change, make sure you document it with photos. Take a picture of your enclosed environment. Take a picture of it at the beginning and at the end, and if there's any big changes that you want to show us, that makes for a really great artifact and really shows me that you took this inquiry project seriously and were interested in it. Complete the lab questions in the Ecology in a Bottle Google Doc and submit it along with any photos, your daily observation notes, and any other artifacts you collected. Anything you've done for this project can be submitted to add on to your grade. Those 10 extra points are going to come in really, really handy, and they're going to boost your grade a lot if you're a student who's maybe in the middle of the pack and you're ready to jump into getting those B's and high A's. So, best of luck to my junior ecologists. I know that you have what it takes to get this done. If you have any questions or concerns, please contact me or Mr. Pomponio as quickly as you can, and we'll get back to you right away so that you can get your project underway and set up. Your project will have to be turned in via Google Classroom on Wednesday, May 20th. That will be the last day we will be accepting this project, so make sure you get it in at least by then or before then if you can. Looking forward to seeing your results. Good luck again, junior ecologists, and I'll see you in the next video. Bye, guys.